Hello, my name is Khan, and my presentation will be about the effect of interferons on alphaherpesvirus invasion and spread in the nervous system. Alphaherpesvirus are enveloped double-stranded DNA virus that can infect humans. Those that infect humans include herpes simplex virus 1 and 2 and varicella zoster virus. Alphaherpesvirus is highly prevalent with approximately 70% of the world population infected with HSV1. This virus is mostly transmitted via oral contact and acquired early in life. Beside the human alpha herpes virus, veterinary alpha HV, such as pseudorabies virus, is a commonly used model to study alpha herpes infection because PRV shares a similar genome structure and replication strategies to that of human alpha herpes virus. It can infect a wide variety of animal models and cell types, and more importantly, it is non-infectious to humans, so it is safe to work with. Alpha herpes virus infection, specifically HSV1, originates at the epithelial sites. The viral particles can enter and travel retrogradely to the cell bodies of sensory neurons in the peripheral nervous system, and there it can establish latency. The virus can reactivate and exit latency, and upon reactivation, the viral particle travels anterograde back at the mucosal site, and the spread event can result in lesions or cold sores. In rare cases, the infection can spread into the central nervous system and cause encephalitis. Currently, the most common treatment includes acyclovir, which inhibits viral DNA replication. However, there is no treatment that targets latency or reactivation. So, even if acyclovir helps eliminate viral replication at the mucosal sites, infection can still occur when the virus reactivates, so it's a lifelong infection. We've talked about possible treatments that can be used to fight off alpha herpes virus infection. What about within our own body? How does the immune system respond to the virus? Upon viral infection, small signaling molecules known as cytokines help alert other cells. And a type of cytokines called interferons are released as intrinsic immune response. And there are three types of interferons. Type 1 interferon includes interferon alpha and interferon beta, which are made by all nucleated human cells for antiviral response. Type 2 interferon includes interferon gamma, which activates macrophages, and it is also involved in both innate and adaptive immunity. Type 3 is a recently discovered type of interferons, and it includes interferon lambda. It is known to modulate immune responses during acute viral infection and tissue inflammation at epithelium, but not much function is known in the nervous system. So, our project goal is to investigate the effect of interferon lambda upon alpha herpes virus invasion and spread in the nervous system. We know that the binding of interferon induces homodimerization of its receptors, and this leads to the phosphorylation of JAK. JAK then phosphorylate tyrosine kinases on the receptor, where the protein called STAT will dock on and get phosphorylated. STAT then dissociate from the kinase as a dimer and enter the nucleus to act as a transcription factor for interferon-stimulated genes responsible for antiviral response. Type 1 and type 3 interferons activate the same JAK-STAT pathway and therefore both elicit antiviral response in the epithelial cells. However, there's again not much is known about the function of type 3 interferons, so it is of our interest to see the effect of type 3 interferons in neurons. To do that, we treated red 2 cells, which are a kind of epithelial cells, and superior cervical ganglia, SCGs, which are primary neurons, with the same amount of interferon lambda to see if there's any difference in the downstream signaling of JAK-STAT pathway. From our Western blot, we noticed that STAT1 is phosphorylated in both RET2 and SCG, but STAT2 is phosphorylated only in rat2. From then, we want to see if the difference in JAK-STAT induction affects antiviral response in epithelial and neuronal cells. So we treated rat2 with interferons before and after PRV infections. Our data showed that upon treating with interferons prior to infection, type 1 interferon reduces PRV yield by 1,000-fold, while type 3 interferon lowers viral yield by 100,000-fold, which was significantly lower. And upon treating with interferons after PRV infection, type 3 interferon is still much more effective at lowering viral yield than type 1, although the effect wasn't as strong as the pretreatment. We went on to compare the effect of type 1 and type 3 interferon in neurons with pretreatment prior to infection. And we noticed that pre-treating SEGs with type 3 interferon is more effective in lowering PRV yield than type 1. And this is to compare the effect of interferons between rat2 and SEGs. In summary, we found that type 3 interferon induces the JAK-STAT pathway differently in different cell types. STAT1 is induced in both epithelial cells and neuronal cells, but STAT2 is induced only in epithelial cells. We also found that interferon lambda treatment has different protective effects in the two cell types. So it could be possible that the different induction of JAK-STAT by interferons leads to the differing protective effect in epithelial and neuronal cells. For future research, we can investigate what genes are transcribed from the JAK-STAT pathway in neurons and in epithelial cells when they're treated with interferon lambda.